discussion about the cell division. As stated in the first part of the cell division has two types, the mitosis and the meiosis. Mitosis is already done and already posted in our first video, so let's move on to the meiosis. The meiosis has two stages, the meiosis 1 and the meiosis 2. The meiosis 1 focuses on the division of homologous chromosomes to produce two haploid cells with duplicated chromosomes, while the meiosis 2 focuses on the division of sister chromatids. Now, let's proceed to the stages in meiosis 1. First is the prophase 1. Prophase 1 is the first stage of meiosis and is the longest phase of the process. Homologous chromosomes from each parent pair up, exchange genetic information and form crossovers. These crossovers are visible as chiasmata. Prophase 1 has several substages, including diplotin, zygotin, and picotin. The prophase 1 ensures that homologous chromosomes are accurately separated during the first meiotic division. This reduces the genetic content of the daughter cells. Prophase 1 also leads to the formation of 4 daughter cells with half the number of chromosomes as the parent cell. And the metaphase 1 is the phase of meiosis where homologous chromosomes line up along the cell's equator. The next one is the anaphase 1. Anaphase 1 is the third stage of meiosis 1, a type of cell division that reduces the number of chromosomes in sex cells. During anaphase 1, chromosomes are pulled apart and moved toward opposite ends of the cell. We have two stages of anaphase, and the first stage of anaphase 1, where fibers connected to microtubules shorten, causing kinetochores to move up or down. And the second stage of anaphase is where the spindle networks elongates, separating the chromosomes further. Here are some other things that happen during anaphase 1. First one is the homologous chromosomes separate. Microtubules disassemble and contract, pulling homologous chromosomes toward opposite ends of the cell. The second one is the sister chromatids remain attached. Sister chromatids of each chromosomes remain attached to one another, maintaining the chromosome's X-shaped structure. And lastly, the daughter cells have half the number of chromosomes. Each daughter cell resulting from meiosis 1 has half the number of chromosomes as the original parent cell. And then the telophase 1. In the telophase 1, chromosomes moves to opposite poles of the cell, and a nuclear envelope forms around each set of chromosomes. And lastly is the cytokinesis, where the cytoplasm of the cell divides, separating the cell into two daughter cells. Now let's proceed to the second stage of meiosis. The meiosis 2 focuses on the division of sister chromatids. Meiosis 2 is the second and final stage of meiosis, and it produces haploid cells that functions as sex cells. Meiosis 2 begins after cytokinesis and resembles mitosis. It involves the separation of sister chromatids, which are two joint copies of each chromosome in the haploid cells produced by meiosis 1. Now let's proceed to the prophase 2. Prophase 2 is the first stage of meiosis 2, a process that creates gametes for sexual reproduction, wherein the chromatin condenses. Chromatin condenses into chromosomes again, and the nuclear envelope breaks down. And the spindle apparatus forms. A new spindle apparatus forms in each daughter cells. And also the centrosome moves to the polar regions, followed by the sister chromatids shorten and thicken. The second stage of meiosis 2 is the metaphase 2. During the metaphase 2 of meiosis, chromosomes line up along the metaphase plate or the cell's equatorial plate in preparation for separation. Microtubules from the centrosome and
The third one is the anaphase 2. The anaphase 2 is a phase of meiosis 2 in which sister chromatids separate and move to opposite poles of the cell.
Centromere splitting happens in anaphase 2, where the centromere of each chromosome splits, and also microtubules from each spindle attach to the sister chromatids at the kinetochore and pull the chromosomes to opposite poles apart. And also, cellular checkpoint occurs in anaphase 2, wherein a checkpoint ensures that chromosomes formed after meiosis 1 have no changes. The telophase 2 occurs after the anaphase 2, wherein the chromosomes move to opposite poles of the cell, they condense, and nuclear membranes form around them. Followed by the cytokinesis, wherein the cytoplasm splits, separating the chromosome sets into 4 new cells called haploid cells. Each haploid cell has one copy of each chromosome. The cells produced by meiosis are genetically unique because of the random assortment of maternal and paternal chromosomes, and because of recombination during crossover. Meiosis is a process that occurs in sexually reproducing organisms and is important for maintaining genetic integrity and diversity. Errors in meiosis can lead to life-threatening outcomes such as Down syndrome, Tau syndrome, and Edward syndrome. That's all for our video for today, and I hope you learned something. Again, this is Tani reminding you all to